And I'm Bradley Wetzell. We work here in the Psychopharmacology Laboratory here at American University.
Today we're going to talk about an autoimmune disease known as systemic lupus erythematosus, or SLE.
SLE is of particular interest to us because research related to it has to do with some of the research that we do here in our lab.
And we'll get to that in a few minutes, but first let's cover some of the basics about SLE. SLE is an autoimmune disease with unknown etiology, but there's a lot of evidence to suggest that it has a genetic component. For instance, if someone in your immediate family has SLE, your chances of acquiring it are significantly increased. Although no single gene has been linked to SLE, the presence of HLA alleles, known as DR2 and DR3, increase an individual's risk fivefold.
Along with genetic predisposition, it's also believed that some form of environmental insult, such as toxins or ultraviolet light, may initiate the first onset of the disease. Additionally, one form of SLE can be induced by certain medications. There are over 400 known pharmaceuticals that can produce drug-induced SLE, but this form of the disease is usually reversible and typically disappears with cessation of treatment.
So what happens when someone has SLE? Well, SLE causes a type 3 hypersensitivity reaction, which is when there are an equal number of antigens and antibodies in the body. In the case of SLE, the antigens are nuclear self-antigens, particularly nucleic acids, but also blood components such as red and white blood cells and platelets.
It's difficult to say exactly where the process begins because it's a circular chain of events that progressively gets worse. For our purposes, we'll start with the fact that SLE patients tend to have a reduced number of tangible body macrophages, which are responsible for digesting the remnants of apoptosed B cells, among other things. These remnants, which include nuclear material and other cell parts, don't get cleared as quickly as they should, and as they build up, the immune system begins to respond to them as foreign antigens.
This results in large quantities of IgG antibodies that respond to these cell components and bind to them to create large immunological complexes. These large complexes can get lodged in major organs, causing organ failure, a major component of SLE. These self-antibodies can also initiate other immune responses by interacting directly with certain epithelial cells and blood components.
Another hallmark component of SLE involves widespread inflammatory responses, because immune complexes that contain nucleic acids tend to stimulate certain toll-like receptors and trigger massive productions of alpha interferon, which is a type 1 interferon. In fact, recent evidence has suggested a possible link between genetic expression of interferon regulatory factors and SLE.
Type 1 interferons trigger an adaptive immune response by activating dendritic cells and causing increased expression of MHC class 1 and 2, chemokines, chemokine receptors, and co-stimulatory molecules. This induces development of Th1 helper T cells as well as cytotoxic T cells due to an increase in APC cross-presentation and inhibition of T cell apoptosis.
Type 1 interferons also promote B cell activation, antibody production, and Ig isotype class switching, and potentiate the effects of gamma interferon and IL-6 to further enhance the inflammatory response. As B cell activation and proliferation increases, the debris from old B cells isn't cleared because of deficient tangible body macrophages, which brings us back to the beginning of the cycle. As you can see, this whole process is a complex positive feedback loop, but the bottom line is SLE promotes and sustains the activation of self-reactive T and B cells, resulting in a large quantity of IgG antibodies that respond to self, create large immunological complexes that get lodged in major organs, and produce inflammation at multiple sites throughout the body.
As you can imagine, these consequences result in a wide variety of symptoms. In fact, SLE is known as the Great Imitator because it is frequently misdiagnosed due to the similarity of its symptoms to those of other diseases. This has led to the development of a list of 11 common symptoms of SLE, at least 4 of which must be present for an SLE diagnosis.
The first of these are a butterfly-shaped rash confined to the cheeks and a discoid rash. This is actually where the name of the disease originates, lupus erythematosus signifying a red skin disease. The other symptoms include photosensitivity, oral or nasopharyngeal ulcers, arthritis in 2 or more peripheral joints, serositis including pleurisy and pericarditis, renal disorder, particularly proteinuria, neurological disorders such as seizures or psychosis, hematologic disorders such as anemia, immunological disorders, and the presence of anti-nuclear antibody.
Currently, there is no cure for SLE, and treatment involves minimization of symptoms with non-steroidal anti-inflammatory medications for mild cases or corticosteroids and immunosuppressants for more severe cases. Newer treatments are currently being studied that involve the use of monoclonal antibodies that neutralize alpha interferon to attenuate the effects of SLE.
This is where the connection to our work here in the psychopharmacology lab comes in. Much of our work here focuses on understanding the aversive effects of drugs that are commonly abused, such as morphine and cocaine, and how the body and nervous system make changes to counteract prolonged exposure to those effects.
One research paradigm that we often use is known as conditioned taste aversion. This is where we give an animal access to a novel taste stimulus that they would ordinarily prefer, like water sweetened with saccharin, but then pair it with an injection of a drug that we're studying so that the animal will associate the effects of the drug with the new taste stimulus. If the drug has toxic or aversive effects, the animal will typically reduce their consumption of the new stimulus over several trials.
What's remarkable about this phenomenon is that the pairing of the taste stimulus and toxic effects appears to bypass cognitive circuits. Many of us can relate to this. If you've ever eaten a certain food while you were sick, you may have had a hard time eating that food again. Even though you know it's not the food itself that made you sick, you still can't overcome that aversion. This is obviously advantageous, and we can see why the ability to quickly form associations between novel tastes and toxic effects would be selected for through evolution.
Because the association appears to be mediated by special neural circuits beyond those responsible for cognition, the conditioned taste aversion paradigm has become an important pharmacological assay for measuring a drug's toxic effects. And back in 1975, two researchers named Robert Ader and Nicholas Cohen decided to use it to test a drug called cyclophosphamide, an immunosuppressant that is often used in the treatment of SLE.
In a normal conditioned taste aversion procedure, water-deprived animals are given access to the saccharin-sweetened solution for a set amount of time over 5 trials. After the first 4 trials, they are also given an injection of the drug being studied, in this case cyclophosphamide. Then on the 5th trial, they are only given the taste stimulus without injections. The difference between how much they drink at the first presentation of the saccharin and the last presentation is used as an indicator of the toxicity of the drug.
But amazingly, when Ader and Cohen gave their rats saccharin on the 5th trial without injections, they died. Thinking that something horrible had gone wrong, they repeated the experiment with the same disturbing results. Further research eventually revealed that each time the researchers injected their animals with cyclophosphamide, their immune systems were compromised, as you would expect, but each injection had an additive effect such that every injection created a more pronounced suppression than the last.
What Ader and Cohen discovered was that the rats had actually formed such a powerful association between the taste of saccharin and the effect of cyclophosphamide that upon the final presentation of the saccharin solution, the rats' immune systems had reacted as if they had received another injection of the drug, even though they hadn't. What's more, their immune system reacted in the same additive manner, and this final suppression had been fatal.
This discovery revealed an important connection between the immune system and the nervous system, but Ader and Cohen didn't stop there. Next, they took rats with SLE and once again paired the taste of saccharin with cyclophosphamide. They found that they could treat the animals with a fraction of the doses normally required for treatment simply by tricking their immune systems into thinking they had received the drug, when in fact they had only received saccharin. By reducing the amount of drug required for treatment, they not only extended the animals' lives, but also improved their quality of life.
More recent research has shown that taste stimuli paired with antigens can even stimulate B-cell production of specific antibodies. The connection between gustatory stimuli and the immune system appears to be mediated by the HPA axis and variations in cortisol levels, but the precise mechanism has yet to be fully characterized.
So that's a quick summary of SLE. We hope that we've given you not only some insight into a serious autoimmune disorder, but also how the study of immunology can affect many other branches of scientific research.